Right, thank you. So my task here is to chair the first session to the break, but also to start off by giving a summary of what the researchers have been discussing over the past couple of days. So there is a health warning with this. I apologize if I've misinterpreted any of the science that was presented or I left it out, but I was frantically making notes throughout. So apologies if I've messed anything up. Okay. So the meeting actually started on Monday, I believe, with the Young Emerge meeting called Sparks ME. There were 50 attendees representing the UK, rest of Europe, Australia, the US, which is an amazing gathering of young people. These are mainly PhD students, postdoctoral fellows, and they are the future of ME research. It's not us older individuals standing up here, they are the future of ME. So it's really great to see them. They had their own program, they made presentations, a lot of it was networking. A lot of the networking involved visiting pubs around Cambridge, and I think they visited the Eagle pub, which is the one that inspired Watson and Crick to solve the structure of DNA. So as supervisors and mentors, we're hoping our students and postdocs come back similarly inspired to solve ME, we can hope. And also Emerge met as well just before the main meeting, and there were two contributions that Emerge made to these meetings. One is to talk about making proposals to the EU, the Horizon program there. They've for the first time identified ME, CFS as a disease of interest in need of more research. So we're looking at how we can make a bid for that funding as well. And then we also talked about, with the contribution from the NIH, on developing clinical trials practices. And this is to develop best practice in clinical trials. And it's been called the Hinkston criteria after its initial foundation here. Vicky Whitmore, with the first presentation, will talk a lot more about that. Into the science program, so day one started with systems biology and artificial intelligence. And I've indicated where the contributors come from rather than put their names. So we had contributions from people in the UK and France. They talked about how to combine huge multiple data sets. Lots of computing power was the sort of outcome of that. And they were talking about taking data from a single cell all the way up to an intact organism, a patient. Okay? Computational and mathematical models are important to make sense of all this data and bring it together. And most importantly, that a lot of them agreed that this sort of approach is ideally suited to understanding ME/CFS and the complexity of the disease. That was then followed by a genomics presentation, and we heard from Decode from the University of Edinburgh that analysis is underway, and we should expect to hear some of the initial findings from that later in the summer. We also heard from Chris Ponting's group that they've undertaken a prevalence study of ME/CFS in the UK, and literally the numbers are doubling from what the original indications were of about 250,000. Now estimates are over 400,000 people affected in the UK. Chris Ponting's group also have been looking at using genetic and blood-based biomarkers, and they're moving on those projects. Again, these will be published in due course. So a lot of very interesting work coming from the genetics. A group from Netherlands have been looking at the receptors on one particular type of immune cell, natural killer cells, and they've identified variants that are specific or associated with ME/CFS. And as many of you may know, dysfunctional natural killer cells is one of the hallmark immune features of ME/CFS. And there's another group based in Austria who are looking for rare variants as a means of subsetting and defining more precisely different clinical subtypes of ME. And that's using genetic databases like Decode and others in the UK Biobank and also American genetic databases. So very exciting developments from genomics and both systems biology. We then followed with a molecular biology focused theme, and we heard from studies being carried out in Canada based on twins, and they've identified increased levels of this particular protein called SMPDL3B, which is important in the fluidity of cell membranes, so making them fluid or more rigid. And these were identified as being higher in the blood of two ME patient cohorts, one in Canada, one in Norway, interested in this protein associated with activation of immune cells as well. And with more interest, perhaps they've identified drugs which can target this particular protein. So this is interesting in terms of thinking about developing clinical trials in individuals which have this elevated levels of this particular protein. We then heard a presentation using cell-based models. So these are cells cultured in laboratory. And they were looking at how rapamycin might be useful to control EBV activation and reactivation, which is, again, is another common feature in ME/CFS. The next session was chronic infection etiology, and we heard from a group this is all based principally on long COVID. There's a lot of parallels and similarities with long COVID, so we're hoping that the information gained from long COVID will provide insights into ME, sharing technology, sharing expertise, sharing the findings that could be relevant to ME. So one study being carried out in Canada, and you'll hear about this later as well in the morning presentation, is they've looked at very large cohorts. They've collected over 1,000 samples, and they've used these to identify distinct clinical subtypes in long COVID. And these symptoms seem to correlate nicely with geography, where the patients live, the environment they're in, and also the SARS-2 variants that they've been infected by. Then there was also LC Revitalize, again from the Canadian group, describing drug repurposing and identifying pathways that could be susceptible or amenable to specific drugs. And there's a couple up there mentioned that target signaling pathways in immune cells, as well as actually cytokines which are inflammatory and cause a lot of damage in the body. And then we heard from an American group, Nancy Klimas's group, who are undertaking a clinical trial in long COVID using an antibody that binds to the SARS-CoV-2 spike protein with the intention of eliminating reservoirs of the virus which may be causing long COVID, which are found in the brain, the gut, and cause neuropathology. So again, lots of things we can learn from long COVID. Then the next session was nervous system and neuroinflammation, and we had talks based on pain in long COVID. There was a mouse model where it showed that they could take antibodies from patients and transfer the pain phenotype in the patient to the mouse. So the antibodies are clearly involved in mediating some aspects of pain and fatigue in long COVID. And interesting, some of these antibodies in the mice directly localized to the brain and to a particular region of the brain called the dorsal root ganglia. We then heard about imaging studies, neuro PET, positron emission tomography, and MRI imaging. And again, they were able to identify that microglia in the brain seem to be continuously activated. And again, they could define different subtypes of long COVID based on the extent and distribution of neuroinflammation in the brain. Then finally, the session was a session from the Netherlands from the brain donor bank, where they've been looking at the hypothalamus pituitary axis. They've had donations from 5 to 9 patients, and they've been looking at the brain, and they've identified some abnormalities in a particular hormone called CRHIR. And again, this links into regulation of the pituitary hypothalamus axis. And these correlate with reduced levels of cortisol in the patient. So that's really novel insights that have been gained through these very generous donations from patients. And then we had an immune system session. And again, this was led by people investigating long COVID. One investigator based here in Cambridge has developed a COVID-19 cell atlas, identifying cells that are affected by the virus, where they are in the body, where they are in tissues. And an important message, I think, that came out from that is that we need to be sampling other tissues and organs other than the blood. So a lot of these cells that are involved in the pathology are found in tissues and not necessarily in the blood. So we thought that was an important message. Then there was a group looking at exercise and immune metabolism in long COVID and some ME patients. They've identified how the immune system responds to exercise, and looking at the mechanisms of PEN, they've identified potential mechanisms that involve changes in microvasculature, mitochondrial dysfunction, immune dysregulation, and then microbiome disturbances which can lead to a leaky gut And all of these could be contributing to PEM. And then a group in Cornell, led by Maureen Hansen, have been looking at cell-free RNA that's in the blood of patients. They've identified a large number of RNA species or transcripts, and these do cluster in case controls, so you can distinguish patients from controls by the clustering of some of these mRNAs. And they're using machine learning to develop models that will allow this to be extended further. Interestingly, they've identified a potential role of a particular type of immune cell called a dendritic cell, a plasmacytoid dendritic cell, which may be elevated in ME. And these dendritic cells are essentially the conductors of the immune orchestra. So they're very important, integral to controlling and regulating the immune response. The next session was orthostatic intolerance and autonomic physiology. And this is way beyond my area of expertise, so apologies to those, particularly David Sistrom, if I've got this wrong, but I understood from the presentations that the one commonality that's been identified is cerebral blood flow, changes in cerebral blood flow and hypoperfusion, so not enough blood being able to get into the brain. And this is a key finding in the ME patients. And then we had a talk from Jesper Melson, and I'm not even going to attempt to explain what the Valsalva maneuver is, but hopefully he will, and the findings from his work. And again, Jesper's now working with mathematical modelers to develop more accessible and user interfaces that can help explain some of the abnormalities in ME patients. And then metabolomics, and this involved contributions from across the globe, as you can see, Australia, America, Europe, and including the UK. And we heard from a talk about the overtraining syndrome as a model of acute PEM, and they were able to identify particular changes in metabolites, and particularly in lipids, which is an unusual aspect. Lipids haven't been really investigated in any great detail in ME. And then there was another study that coming from Canada, that involved Canada, and again they saw a similar elevation in certain lipids in the blood. So lipids may actually be quite a key player in some of the pathology related to ME. And then we had a presentation from the US, James Baraniak, who's for a long time been looking at Gulf War syndrome, and he's been looking at that as a means of finding clues as to the causes and ways of treating ME. Interestingly, he was talked about a cancer therapy called CART, which targets T cells in cancer therapy, and a consequence of, or a side effect of the treatment is the development of cognitive issues. And this is manifest in terms of microglia activation, changes in key structures and cells in the brain, and this leads to neurocognitive dysfunction. James thinks there could be an important clue here for ME, and that one of the key players in ME is this ion channel called TRMP3, which we've heard about in several colloquia. And this defective ion channel, when it is defective, and it appears to be defective in ME patients, leads to a leaky barrier in the blood-brain barrier, allowing potential toxins, etc., to get entry into the brain, which of course could then lead to cognitive deficits. So that's a very exciting development as well. And then there was an exercise test, stress test in ME, Gulf War and controlled individuals using proteomics and looking at messenger microRNAs. Found no differences in proteins, but interestingly there was a small number of microRNAs that seemed to be a signature in the ME patients. And this is linked to changes in serine levels in the cerebral spinal fluid of ME patients. So again, important potential biomarkers that could be of use for ME. Then we had an interesting talk from somebody based in the UK who's using flies, Drosophila, as a model of aging, and which again may be related to ME because we believe that accelerated aging or senescence is an important feature, particularly of the immune system in ME. And what she described was but increasing catalase production actually increased life and health span. So looking at the redox potential, which is abnormal in ME patients, could actually be a useful therapeutic target. And then we had metabolomics and invasive cardiopulmonary exercise testing. This was from David Systrom in Harvard, who's going to be talking about this later in the program. And he identified abnormalities in oxygen delivery to the heart and utilization. And this was followed by a talk from the Netherlands where they've actually identified microvasculature changes in the muscle, skeletal muscle of patients. And this actually may be contributing to some of the effects in the heart. So nice link-ups there. So impaired oxygen delivery, which impacts on cardiovascular function. And this is the last slide. So the very last session was, this is new to the colloquium this year, was getting presentation from people working with in vitro and in silico models, which could be of use in ME and understanding the disease etiology, or in fact be used in drug testing. So we heard from Elisa Ultra in Spain about inducible pluripotential stem cells and how they could be used. In Norwich, we're developing organs on chips or body on chips to look at interconnectivity of different organ and cell types using patient material. And then presentation from Imperial College using organoid systems. So there are lots of advantages to these models. So it overcomes a lack of suitable patient samples because they can be propagated in the laboratory. We can study many cell types with these systems, allow sharing of samples, and they can be genetically manipulated to change or knock out specific genes we may be thinking that contribute to ME. And so it allows studies of disease causality as well as drug testing. In silico models, so this is all computational, AI, network biology, systems biology, all of these things, allows the connecting of multi-source data, so multi-source data that's available freely or from research groups. It allows pathway discovery, and it can identify potential mediators of disease processes, and these sort of interactomes, how different organ systems and interact with each other. And importantly, they can generate hypotheses which we can test in some of the other in vitro models before we go into patients as well. There are, of course, drawbacks with this, and we're all working on these. So at the moment, it's very expensive to do. Technically, it's quite demanding, and we have no SOPs. We're all working on these to try and make it more accessible and user-friendly for ME researchers. So over 2 days, I think just what I presented here, I hope you can get an appreciation of the fantastic science that's going on in ME, and a lot of things seem to be moving forward. There's a lot of commonality coming up, some key markers that could be of use diagnostically as therapeutic targets. So I think the next couple of years offers an exciting opportunity to put some of these into the clinic and actually to be benefit to the patients. So that is, I think, my last slide. It is. So yes, enjoy the day. So my last task is to introduce the next speaker, and this is Vicky Whittemore from the NIH in the US. Vicky has been absolutely integral to trying to get this consensus on how we design and improve clinical trials in ME/CFS patients. And so she's going to talk about clinical trial standards, which are emerging from this group of Europeans, Americans that are coming together, mainly clinicians, that are trying to standardize clinical trials so we actually know what we should be measuring, how we should be measuring, and what it might mean in terms of clinical efficacy. So, further ado, I'll hand over to Vicky.